Good night. My name is Aranza Valencia, and I'm going to talk about camouflage in cephalopods, mainly in the cuttlefish. Cuttlefish belongs to the phylum Mollusca, the class Cephalopoda, and the order Sepida. They are characterized by having a soft body, but also by the ability to develop camouflage. But what is camouflage? Camouflage is known as the adaptation that allows animals to blend in the environment. These adaptations can go from simple alterations of color to complex texture changes. The main function of camouflage in cephalopods is as a defense mechanism, but in some species is also used for sexual selection. In cephalopods, camouflage is controlled by the brain. This is the reason why they can change their pattern in less than 1 second. The keys to camouflage process are the chromatophore organs that are in their skin, the structural cells, and their vision. Chromatophores in cephalopods are considered neuromuscular organs instead of cells. This organ is constituted by 5 types of cells. The pigment cell is characterized by having a flexible sac that contains a large amount of pigment granules. These granules can be yellow, red, orange, and brown, or they can also be black, and they depend on the kind of species. These cells are surrounded by 10 to 30 muscle fibers that are radially arranged. They communicate between each other through stimuli, produced by neurons that can transport different signals from one side of the body to another. Along the radial muscles, there are branched nerve fibers that contain nerve bundles with 2 or 4 axons. They also have a series of transmission sites called synapses that make a connection between neurons and the muscle fibers. Nerve axons are always accompanied by glial fibers. These cells fulfill the function of covering the outside surface of the axons as they move along the muscle fiber. Finally, the sheath cells have the function to cover the pigment cells and the radial muscles, giving them the support that they need for camouflage development. In cuttlefish, there can be found 2 types of structural cells, the leucophores, which have the ability to diffusely reflect all the wavelength of light in one single time. As a result of this, a bright white can be produced, and the iridophores, which are more complex. They contain a protein called reflectin, which bounces only a specific wavelength of light back out. Their vision is characterized by having a large and complex eye, huge optic lobes, polarization vision, night vision, but also they do not have color vision. It was already explained the main factors that allow camouflage to develop. Now we will actually see how this mechanism works. It all starts with the vision. They use their eye lens and optic lobes to analyze what is around them and get an approximation of their environment. Once they have their target, the optic lobe sends signals to the basal lobes, and after, they transmit these signals to the chromatophore lobes until the chromatophore organ. As these signals arrive to the chromatophore organ, the radial muscle fibers contract, shrinking the pigment sac and allowing the structural cells to reflect the wavelengths. To make a match in the color and pattern, they evaluate the intensity of the light reflecting in the background, the size of the pattern, and the properties of illumination. This whole process can be developed in less than a second. In addition, Some species can also use their arms to mimic animals or seagrass. To conclude this presentation, it can be summarized that there are three key aspects in the development of camouflage. The first one, and the most important one, is the vision. The second one, the chromatophore organs, and the third one, the structural cells that are attached into their skin. To be able to process these mechanisms, cephalopods might have passed through a complex evolution process that made them one of the most remarkable animals. Even if there is a prolific amount of research in this process, there are a lot of aspects that should be discovered. Thank you.